Hello everyone. Today we are going to discuss variations of PCR. In the previous sessions we already learned the concept of the PCR and the concept of the primer design, etc. Now, as a widely used technology in molecular biology and genetic engineering nowadays, the PCR technology has evolved in different directions to suit different applications, and variant forms of the PCR technology has already developed according to the specific needs of the scientists. And now we will see one by one what are the different types of PCR technology that is available. This is the list of the PCR technologies or methods that is nowadays available. The first one is the inverse PCR, then hot start PCR, multiplex PCR, nested PCR, quantitative PCR, which is also known as qPCR, reverse transcription PCR, which is also known as RT-PCR, touchdown PCR, asymmetric PCR, allele specific PCR, then assembly PCR or polymerase chain cycling assembly. That assembly PCR or polymerase cycling assembly, that is PCA, then digital PCR, also known as dPCR. So these are all the different variants of the PCR technology that is nowadays available, and we will discuss it one by one. The first one is the inverse PCR, and the inverse PCR is a commonly used technique, and it is commonly used to identify the flanking sequences around a genomic insert. It involves a series of DNA digestion and self-degradation, resulting in known sequences at either end of the unknown sequence. So our main target is to amplify an unknown DNA sequence. Why you want this inverse PCR? Because generally for every PCR technique we require a primer, which is usually constructed or designed against a known sequence. But without the knowledge of the prior knowledge on the sequence of the target DNA, we cannot construct a primer. So an unknown DNA cannot be generally amplified using such a PCR technology, because we cannot construct a primer against an unknown sequence. There comes the role of the inverse PCR, where we will use a non-DNA sequence using using RDNA technology. We will insert this non-DNA sequence construct into the unknown sequence. So in between that unknown sequence, which is usually it is the flanking sequence on both sides. So this unknown flanking sequence on both sides is now present after the incorporation of this non-sequence in between. And a restriction digestion can be done. Then fragments can be separated, and the fragment with the non-sequence can be purified, which can be self-ligated to form a circularized RDNA construct. And now your unknown sequence is circularized in combination with that non-sequence. Usually in the non-DNA construct that is inserted in between these unknown DNA, there will be a restriction site. And if you are targeting that site with a concerned restriction enzyme, the circularized DNA construct will now be linearized, and that linearized DNA will have such a structure in which on both ends we will have the non-sequence, and that non-sequence can be used to construct a specific primer against that sequence. So the entire segment, including the unknown DNA sequence in between, can be amplified, and that can be amplified, and even that can be used for a sequencing of that non-DNA sequence. So this is the inverse PCR. The next one is the hot start PCR. Hot start PCR is a technique that reduces non-specific amplification during the initial setup stage of the PCR. It may be performed manually by heating the reaction components to a denaturation temperature, that is 95 degrees Celsius, before adding the polymerase. So a non-specific amplification before the starting of the cycle, that is zero cycle amplification, is an issue. because that will be non-specific. Even if the reactants were already mixed, and the PCR start machines, so the PCR reagents, where they starts working even before the starting of the cycle and concerned temperature is reached, then non-specific binding and amplification can occur, and that needs to be prevented. If you want to prevent that non-specific amplification, then hot start PCR is the solution, where you will that can be performed by heating the reactant mixture components to denaturation temperature, that's 95 degrees Celsius, before adding the polymerase. So the polymerase enzyme is added only after reaching the 95 degrees Celsius annealing temperature. So before that, you are not adding the polymerase. So non-specific amplification at lower temperatures will not occur. Specialized enzyme systems have been developed that inhibit the polymerase activity at ambient temperature, either by binding of an antibody or by the presence of covalently bound inhibitors that dissociate only after a high temperature activation step. Hot start or cold finish PCR is achieved with new hybrid polymerases that are inactive at ambient temperature and are instantly activated at elongated or high temperatures. So there are specialized hybridized polymerases other than the standard ones, and also there is a some other methods in which it is activated only at high temperature, not at ambient temperature. So this is an illustration of hot start PCR, in which the template DNA is represented here. The tag polymerase, normally it is not able to bind and start the PCR amplification. And as you know, magnesium ions were required for these tag polymerase binding and amplification. But magnesium ions were normally bound with in case of a hot start PCR system, it is bound with pyrophosphate. So magnesium is not free for the tag polymerase to use it and start with. So at ambient temperature or at room temperature, the magnesium ions were trapped with the pyrophosphate. So tag polymerase cannot use them and start the PCR amplification. But when it reaches about 70 degrees Celsius or more, the pyrophosphate is activated. The pyrophosphatase enzyme that is activated, and pyrophosphate will be cleaved by the pyrophosphatase, which will release the magnesium ions. So once the magnesium ions were free, the pyrophosphate is cleaved and magnesium is released, and at 70 degrees Celsius, the template DNA will unwind, and the tag polymerase is now free to bind with the magnesium ions, and at that temperature only this tag polymerase and magnesium ion in combination, it will bind with the template DNA and start the polymerization process. So this is how the hot start PCR works, and at lower temperatures this will not occur because pyrophosphate is actually bound with magnesium ions, so it will not be free for the reaction to start with. So the next method is or the variant of the PCR is the multiplex PCR. Multiplex PCR, actually it consists of multiple primer sets within a single PCR mixture to produce amplicons of varying sizes that are specific to different DNA sequences. So the template DNA may be a very long one, or there may be multiple template DNAs, and you will use, instead of a pair of primers, that is a forward and reverse pair, you can use multiple primers, multiple sets of the primer, forward and reverse primers, so that it will amplify its own target sequences. And in the single reaction mixture, instead of getting one PCR product, you will use multiple primer sets, and each primer set will amplify its own target sequence in the same reaction mixture. So a multiplex PCR is the simultaneous amplification by multiple priming in the same reaction mixture. By targeting multiple genes at once, additional information may be gained from a single test run that otherwise would require several times, and the reagents is required in each and every step, and more time and reagents were required if we are doing it separately. But that can be avoided by running all these things in the same reaction mixture. So you save time and reagents and other resources. Annealing temperature for each of the primer sets must be optimized to work correctly within a single reaction. So there is a proper standardization of the multiplex PCR system is required, and it is based on the nature of the primer and the amplicon size. Everything needs to be considered while designing a multiplex PCR system. That is, their base pair length should be different enough. That means when we are talking about the amplicon size, their base pair length should be different enough to perform, different enough to form distinct bands when visualized in gel electrophoresis. There are two types of multiplex PCR: single template PCR reaction or multiple template PCR reaction. And in the last paragraph, when we discussed, when we explained that these multiplex PCR is actually dependent on the nature of the primer or the different and the quality of the primer, and also on the amplicon size. I will explain it a little bit. Means you know that the primer quality depends upon the GC content and its TM value or the melting temperature and other properties, overall length of the primer. All these affects the TM of the the annealing temperature of the primer, etc. So these all varies. So when you are designing such a multiplex system, different primers that may have different length, different TM value, different GC content. So the primer combinations that you are using in the mixture may be different. So you have to optimize this entire reaction so that all the primer pairs should work at the same annealing temperature. For example, for one primer pair, 62 may be the optimal annealing temperature. Another primer pair, there may be 58 annealing temperature, and for another one, maybe 56 optimal annealing temperature. You cannot use all these different primer pairs which will have different optimal annealing temperature in the same reaction mixture. So in that case, you need to select such primers which will work on the same annealing temperature so that it can be placed in the same reaction mixture and amplify in the same conditions. And also it depends on the amplicon size. Amplicon size, for example, if we are amplifying different target sequences using different sets of primers in the same reaction mixture, after that you will analyze the products on a gel electrophoresis. Each and every gene product of the primer set 1, primer set 2 and primer set 3 can be distinguished. It should be distinguished based on their sizes. There is no other option. So when you are running it in a gel, there should be 3 separate bands available so that you can verify the amplification product of each and every primer pair based on their difference in the length. If all the primers were producing same amplicon size products or the same size amplicons, then what happens? You will get only one band. You cannot differentiate between the products of the primer set 1 and 2 or primer set 2 and 3, etc. Which means you cannot verify whether the concerned gene is present or not, or whether it is amplified or not. It cannot be verified from the gel electrophoresis results. So for an optimal multiplex PCR reaction, you have to select such primer pairs which will have some same annealing temperature and similar properties, similar reaction conditions. Then only it will work in the same reaction conditions in the same vial. And also their amplicon sizes of or the product lengths of these primer sets pairs should be different so that in the gel electrophoresis they can be visualized as separate bands based on their molecular size. Otherwise, the bands will mix up and you cannot differentiate between the products of two different sets of primers. That's why in a multiplex PCR system, always the amplicon size of the different primer sets should be different. You have to select the primer pairs in such a way. And there are cases where you will use multiple primers against a single template, or you can even target the multiple templates with the different primer pairs. For example, say in the single organism, or for example, you can extract the DNA from E. coli and target 3 different genes, maybe ampicillin, tetracycline, or some other different genes using 3 different sets of the primers and see whether it is present or not. That is an example of single template PCR amplification. Whereas if you have a mixed culture in which you extract the entire DNA, that culture may be presented with E. coli, Pseudomonas, Salmonella, or 3 or 4 organisms. You can design specific primers against E. coli, Klebsiella, Pseudomonas, or Proteus, maybe. 4 different organisms can be simultaneously amplified. That the specific DNA sequence can be simultaneously amplified, and you can see in a mixed culture, or for example, a water sample or some other environment sample, the DNA is totally extracted, and you will target that DNA sample or multiple template DNA sample with concerned primers. And if the sample is presented with such template DNA, for example, one sample may have E. coli, Klebsiella, and Pseudomonas, and not Proteus. So there should be 3 bands which will be corresponding to the 3 different organisms that is present, and there will not be any band for the Proteus. And in another sample, maybe E. coli may not be present, but the other 3 organisms may be present. The corresponding band will be visualized. So your target may be a single template reaction or a multiple template reaction. And in single template reaction, the same pure culture organism may be targeted with different sets of primers for different genes. Whereas in multiple template reaction, you can have a mixed culture with different sources, and in that mixed DNA sample, you can use different primers so that in the mixed culture you can identify the presence of different organisms or different template DNA. So that type of multiplex reaction is very useful in saving time and reagents and reagent costs. So this is a one illustration in which, for example, strain A of the organism, which the DNA is extracted. Usually, it can be amplified, and strain B will have its own DNA, and that using specific primers you can amplify. Strain C, using its own specific primers, you can amplify. Strain C, and the DNA can, so in a mixed reaction, for example, all the samples, that may be a mixed culture. For example, water sample with the organism A, organism B, organism C, and organism D. When it is extracted, the concerned DNA will be present in the same mixture, and that DNA can be amplified with the concerned specific primers for organism A, organism B, organism C, and organism D. So in the same reaction PCR vial, we have all the different template DNAs from different sources, and the corresponding primer is used in the same reaction mixture. And after the amplification, if you run in a gel, this is the molecular weight marker to identify the molecular weight of the band. And if all the samples, all the DNA from 4 different organisms were represented in the sample, all 4 products will be obtained. As I mentioned, these PCR products should have different length. So this one is the lightest band with 141 kDa, and this is a 141 base pairs. This is a 202 base pairs. This is a 275 base pairs, and this is a 371 base pairs. So based on the molecular size, it will separate, and the lowest band will be on the bottom, and the heavier band will be on the top. So this way, in the same reaction vial, you can identify different target sequences in the same reaction mixture. That's the concept of the multiplex PCR. So here is another example in which, for example, we have the DNA which is represented here, that can be amplified, and using different primer sets, it is amplified. And for example, this DNA is amplified using another one set of primer, and this DNA can be amplified in another using another set of primer, and another DNA can be these three different DNA may be from three different organisms. You will use specific primers against organism 1, organism 2, and organism 3 in the mixture. And each primer set will amplify its own products. So the red one represents one primer product, and the blue one may be another product, and the next one may be another product. So all the products will be amplified in the same reaction mixture, and that will be separated in the gel during the electrophoresis. So you will have three different bands. And the trickier part is that you have to select the primer pairs in such a way that they will work in the same reaction conditions, so that all the amplification products will be obtained in the same annealing temperature and reaction conditions. And also you have to take care that the primer pairs were producing amplicons of different lengths, so that they can be resolved in the gel at as specific bands or separate bands in the gel. And if two genes were producing same length of products, both the bands will merge together and you cannot differentiate between organism A and B. So that is the issue. So this is about the multiplex PCR. And the next one is about nested PCR. The nested PCR actually it increases the specificity of DNA amplification by reducing background due to non-specific amplification of the DNA. Two sets of primers are used in two successive PCRs. The first step reaction is one pair of primer is used to generate a DNA product, which besides the intended target may still contain non-specific amplification of DNA fragments. So in the nested PCR you are using two sets of primers for a PCR process, PCR reaction. And in the first step reaction you are using the first primer pair which will amplify the target. Maybe there is some part of non-specific amplifier, some amount of non-specific amplification may be expected, because PCR, nested PCR is actually intended for increasing the specificity. But non-specific reactions may occur if the primers were not so specific. So after the amplification with the first primer, you will have PCR products amplified, but with some amount of non-specific amplification products. Then you use the second set of primers which will work inside, in the inside region, and that results in the production of more specific bands in the PCR reaction. So the products are then used in second PCR with a set of primers whose binding sites are completely or partially different from and located 3' of each of these primers used in the first reaction. Then the nested PCR is often more successful in specifically amplifying long DNA fragments than conventional PCR, but it requires more detailed knowledge of the target sequence. You can see some representations here, like this is the target DNA that needs to be, or template DNA that needs to be amplified. And the first round you will use a pair of primers which will amplify the region in between these two primers. So you have the first amplification product. But you are expecting some amount of non-specific amplification in this one. So this amplification product may contain some non-specific target sequences also. So there comes the second step in which these amplified products will be amplified using the second sequence. Actually, your target is this red region. This is the region. So the first step reaction is done by designing a primer against some of the flanking regions on both sides. So after this amplification, you will have the amplified products with maybe some amount of non-specific amplification products. But when you use the second primer, this will bind more specifically to this region, and you will get the gene of interest amplified. So this second step primer ensures that only the concerned target DNA is amplified, and if there is any other non-specific amplification product, that will not be amplified in the second primer reaction. So this is how the nested PCR will work. The second step amplification is ensures that there's a two-step verification process, and if some non-specific amplification is occurring under the first primer, then the second primer ensures that non-specific amplification products were excluded.